Okay, so for the paper chromatography lab, we had two purposes. One was to identify if the dyes were made up of only a single color or a mixture of colors, more than one color, all that. So this would be the red, green, blue, and the Kool-Aid colors. Yeah. And we would do this by identifying the individual colors in the dye and finding the R of F values for evidence. Yeah. And number two is to find how well the dyes moved along the paper by measuring their distance that they traveled after the paper dried. All right, moving on to methods of work. So, like most of the class, we kind of fumbled through the entire process, but I'm gonna get to our problems really soon. So first, let's address the methods of work. So, methods of work. We cut the circular paper into rectangles because that would be the best way for it to fit into the beaker. And yeah, after going through the entire procedure, we pretty much did the procedure verbatim. But the day after we did the experiment, and drying the strip, we measured the distance that the dyes traveled. So our problems, which is a lot of them. Okay, so both of us, we did the pre-lab extremely poorly. That was definitely our fault, but we would definitely do the pre-lab more carefully next time to fully understand the lab. And we were a little confused for most of the process, but we managed to figure it out along the way. I mean, we don't have great confidence about what we did, but from my memory, we went through the process pretty verbatim. Okay, next, we waited too long to check on our strips after putting them into the water. Yeah, we waited too long to check on them after they dried, and we couldn't see the solvent line super clearly. Actually, it's kind of non-existent. Yeah. Alright, so number three, our problems was the beaker that well, I'm not really sure if this is really a problem, but this was definitely not according 100% to the methodology. So the beaker that we used was much larger than what was specified in the instructions, which was a 400 milliliter beaker. And there probably shouldn't be any effect on the results or anything, but just decided I should probably say that. And definitely just read the instructions more carefully, because then we could have more confidence about it. We should have organized it better and just done the pre-lab much better.
During our experiment, we found that the Kool-Aids traveled less than the dyes and that one dye and one Kool-Aid were mixtures. Using the colors the dyes separated into, we found that the green dye and the Kool-Aid 3 were the mixtures. The Kool-Aid appeared to be a mixture of red and yellow dyes, while the green dye appeared to be a mixture of blue and yellow dyes. The RF value for the Kool-Aid mixture was 0.55, and the RF value for the green dye was 1.00. As you can see, the distance the Kool-Aid traveled was less than the distance the dyes traveled, showed by both a lower RF value for the Kool-Aid and that the distance the Kool-Aids traveled was less than the distance the dyes traveled.